modeling RNA, the RNA complexes using different computational approaches. So now I will share my screen, and just 1 second, please. I just want to do one small stuff. Sorry, just before that, I want to, since I'm not so used to this kind of platform, I just wanted to add my images so you can see me and maybe also my hand when I try to explain you what I'm trying to tell you. So let me do that. So during this presentation, I wanted to tell you a bit more what we are doing, and in particular, in our lab, we are positioned in Paris, as was said, and we work on different kinds of topics that spans from X-ray crystallography and MR to try to demonstrate structure, as well as we work on medical chemistry, and we try to put all that together using molecular modeling and bioinformatics. So why we want to study, what is the main object of our work is to look at the cell complexity, and in particular at the crucial role of biomolecules and biomolecular interactions. And to do so, in fact, if you look at the molecular machinery on the cell where planted that also are related to the fact that biomolecules interact. But to have insights on that, we need to know the structures. And if you now looked at the structures and how many PDB structures are available in the literature, we realize that I put these numbers on 2023, but the number is just slightly increased, that if we look at the whole PDB, we have around more than 2,000 structures. But these are mostly proteins. And if in the country we start to look at what it is, protein RNA, protein RNA and the RNA, they represent just a small portion of this of the PDB, meaning that there is a lot of difficulties that can arise to determine that. And why there is these difficulties? There is also other key players like the conformational changes that can occur, that can bring to different conformational changes at the level of RNA. And even though we have this kind of information, if you look at the cell, there is the water molecule and the crowding that can have a big impact. So my research activities are mostly dealing with the three main topics. One is RNA integrative modeling and RNA dynamics using chemical probing data. I will show in a minute what I mean. The prediction of conformational changes, since RNA and proteins can undergo large conformational changes, and even more, we can characterize the protein-RNA complexes and interfaces and dynamics using some specific approaches. And to do so, as you can imagine, we can use different computational approaches at different scales. So the scales that I use to work are mostly free. I work starting from the top with the coarse graining approach that allows us to have pieces of information, for example, of the mechanical properties, and we can also compute a normal mode analysis. If you are used to normal mode analysis for proteins, we can have that represent just the each nucleus residues with C alpha. But we can also go down and use more molecular mechanics to determine the dynamics at a or atom level. And then if you go really down, we can have quantum mechanical calculations where we can compute, for example, the reaction that occurs at the chemical level. We can optimize the structure, but we can also bring together the two region, the two-level theory, the QMM calculation and the MM calculation to try to have on one side the flexibility of the structure and also the reaction that then can place at the QM level. So when we look at the RNA is a very unique molecule because if you start like in a perspective of the proteins, we can have that we have a one structure, so the sequence. Then we can have a 2D structure that are plenty of secondary structure from the level of the RNA that can then interact between each other by forming 3D contacts that bring to a 3D structure. And this, when we look at RNA, we have a plenty of different foldings that can occur, but we have to remember that each structure represents a specific function. So there is a lot of variety on the structure that we don't undermine and we don't understand sometimes. And even more, when we start to look at what are the possibility to form some interaction also inside the RNA, We know that for DNA, we are used to this Watson-Crick basis. On RNA, we don't have just that, but we have plenty of diverse possibility and combination that can occur because we can take an account of the different ages, for example, the Unison ages, the Watson-Crick ages, and the sugar age. And at each age, we can form an interaction. Bringing that, we have 288 possible pairing at radical level that brings the fact that it's a very complex molecule. And also, although we don't all of that were found experimentally, we have up to 145 pairings that are also observed in the NDB, that is the analogous to PDB for nucleic acid. This brings to us that it is quite complicated to determine the structures, although we have high resolution techniques like NMR, X-ray crystallography, RBM, that based on the fact that we have this pairing that is can be different. Also, the ribose that with respect to the ribose is much more reactive and flexible. We have several limitations to determine the structure. So people to try to determine the structure, but decide to do a step back and to look at the 2D structure. And to do so, people look at some thermodynamic model. They consider only canonical pairing at first stage. We use not to use pseudoknot. Pseudoknot is a specific feature of a of RNA that is 3D contacts, where we have an interaction between the loop, for example, of a stem loop, and a completely not per region. And this is a really particular type of interaction that is very famous for RNA for stability. But and there is several techniques, several methods that try to minimize, based on this thermodynamic model, the energy to obtain the secondary structure. Although this is quite important, there is a lot of limitation, because here on the left I show you the example of a prediction RNA structure, and the right the X-ray structure, and we realize that the structure are completely different. There are some regions, in particular, it's the structure that are quite good predicted, but we have a lot of part of the RNA structure that are not completely solved, and so brings the fact that we need maybe some validations in the predictions by mutations analysis and also on phylogenetic studies. But there is also another way to try to solve this problem, is to use some low resolution data. And this low resolution data, how we called in our RNA community, is the chemical probing techniques. This chemical probing techniques allows the probing of the RNA on some specific chemical function that can be the hydroxyl or the ribose or other nitrogen or other part of RNA. And why is it important? Because doing this kind of approaches allows us to have plenty of data. The actual amount of data of chemical probing is order of magnitude larger than the PDB structure available, but are even less, are really few, because they represent less than 1% of the PDB structure. So to do so, one of the key techniques is called shape. That is a technique that is quite important because it allows to have an hydrosylation of the ribose, and there is different probes that can occur to do that. And this is a quite peculiar techniques, because based on the different probes, we can have different informations, but all of them has the on their course the fact that we have an acceleration, and this acceleration occurs at the level of ribose. And this reaction, although it's quite common and quite used, still has a lot of part of this chemical reaction that is not understood. I just wanted to clarify that shape, what is the acronym, because I didn't say it, stands for selected supreme hydroxyl acylation analyzed by Prana section. So this reactions allows to have information at nucleotide level. So in principle, if you have here I show something that is quite common, that is white is meaning that it's not reactive, yellow that is medium reactive, and red high reactive. And from the predictors of the 2D structure, they use a set of constraints, meaning that if it is not reactive, meaning that it forms canonical pairing, although on the contrary, if it is reactive, it should not pair. If you put this kind of information on a 3D structures, we realize that it's not always true, because there are regions where there is paired that are reactive and regions that are not paired that are not reactive. So why is that? And so several open questions arise. And this is like when I started to work on that, I felt like in a laboratory where I could not find my solution and my message, because there are plenty of things and plenty of factors that I could not understand. So the first step to try to understand this system was to investigate the interplay between flexibility and mechanical reactivity using these simulations. And to do so, we set up a benchmark, and we computed different geometry parameters like distances, angles, hydrogen bonding, also packering, that is the angle at the end of every bond, to try to have some pieces of information. And this is the type of systems that we studied are large RNA with different sites. And here I use an arc representation, meaning that each arc represents RS2 nucleotide that form a canonical pairing. So based on that, we set up a statistical conformation analysis by starting to validate that our structure were really stable. So we can assess also, we started their average and their fluctuation and their flexibility. So we verified that the hydrogen bonds were stable along with simulations. Then we compute some averages, some fluctuations, and we do some classical correlation to try to see the association, the strength of association. Even more, we look at multiple correlation coefficients to try to see if more than two quantities can be correlated together and they can bring some pieces of information. And since from experimental level, we divide the reactivity on low, medium, and high, we decided also to see if there is an association between classes, between high reactive and high flexible, for example, regions. And to do so, we use a primary test. So first of all, by look at the correlations, we realize that there are some correlations that are medium-high importance, but nothing is really predominant. And also, since we also think we thought that the hydroxyl is and should be exposed to be rapid to have a reaction to take place, we thought that there was some kind of correlation between the accessible area of this region and the reactivity, but no correlation was found, and this was also confirmed by other people, other researchers, literature. But we saw also at the same time that it was an improvement of the correlation coefficient with two more parameters. Also, since variables is the key element where the reaction takes place, we decided also to compute the packering, meaning that in the DNA, because maybe you're more familiar with that, there is a specific conformation variables that is called C2' end conformation, and to RNA this conformation is the blue one, the C3' handle. And this is mostly represent more than 90% of the conformation variables in the RNAs. But when we compute here red, the probability to find the other packering that is less predominant in the RNA, we observe that on the contrary, it is overrepresented when the reaction takes place, meaning that there is a crucial role of variables and its flexibility in these reactions. Either more, since we wanted to see if there was some, we want to be able to actually take place, we should have some specific group, chemical group to be close to variables to help to the protonation, to help the reaction to take place. We realized that when variables is this not common conformation, we observed that, on the contrary, the distances between the different chemical groups is very high, so meaning that we maybe have an enhancement conformation due to this packering conformation. To go further, since we want to also understand better why on the 2D prediction also fails mostly when we also had shapes data, we wanted to look if there was some association between the packering and the reactivity and the packering and the number of hydrogen bonds that can form, and realize that from if one side the verbose flexibility has a medium association with reactivity, a very weak association is present when we talk about the probability of the forming of the verbose to be in a specific form and the number of hydrogen bonds, meaning that there is not a trivial relationship between the hydrogen bonds and the verbose that is the crucial player in the reaction. So So we thought, and we think until now, that we need a stricter and more sophisticated approach to take in account shape data also in the predictions in the structure. So we want to go further, and in our team, we do also new experiments. We look at the dependence of the temperature and of the chemical probes, and we observe that different activities can be observed based on the probes we use. So based just on the nuclear facility, the properties of a probe, and also the temperature. Meaning that there is a region where, if you look here, the plot, like which decide to classify a nucleotide based on the reactivity changes. And we observe that, for example, the blue one, we have a change of reactivity just occur very late, because that is very constant along the temperatures, meaning that there should be some regions that are highly paired, like nuclear helix. And on the contrary, there is regions that change temperature, reactivity along the temperature. And this, we thought that this can be due to the fact that can form also some thermic contacts that can be quite important. And so we thought that this data, if implemented correctly, can bring also us to have a prediction of, for example, thermic contacts that are really complicated to do in the modeling of the RNA. So we also look at some MD simulations again to see if there is some specificity on the system, and we realize that there is changes of formations and changes of activity in regions that has not standard properties like quadruplet, triple helix, and also no standard cycling, meaning that, again, the nucleotide and their conformation and their flexibility are crucial in this stage. But what we have done with just now was not to take into account explicitly our probe. And now, so we decided the project was founded in 2022, that we should take into account finally the probes, as was shown by the group of Professor Bussi in the past, and where we just look at some specific binding of the RNA. And so we decided to want to compute a systematic study where we do standard simulations, we define new geometric parameters, and we wanted to take all these stages, take into account properly the probes, and also to compute the binding for energy, because it's the first stage before reactions took place. And all this piece of information could be the starting point then to a deep learning approach to predict the shape of activities, because we wanted to find what new descriptors are allowed to be relative to the activity of RNA, but in this case, we wanted to understand how also the probe influences RNA. So to do so, we started with a model system that is SIM1 riboswitch, that is a well-known structure for which we have plenty of PD structure, plenty of data in our lab, and we started with one bias on lateral deep simulations, 1D and 2D on lateral umbrella sampling, and QMM calculation to do reaction. And to do so, we have different reactivity profiles to try to have the prediction of the chemical probes and conditions. So first of all, we verified that RNA is stable along the simulations, and also we decided to take a specific alpha loop since it's not interact with the rest, so it should be independent to what's going on the rest of RNA. So we have experimental data. Today we're just on the senile in the left, and we have data that change the concentration and change also we have a nucleotide is not reactive and two nucleotides that are medium and are reactive. So we did first of all in the simulation just take into account an umbrella sampling, so meaning that we put a bias on the distance and we computed the free energy, and we had to observe that first of all, for one nucleotide that's not reactive, it's not possible to have a binding for energy. On the contrary, when there are free nucleotide, we are able to have a binding that are 1 kilocalorie per mole, that is quite good, but we cannot distinguish between the high and low and medium reactive nucleotide. And also we look at what's going on around very nucleotide and realize that there are different possible conformations that are not some distance that is important, but also some angles. So why don't take into account that in our simulation? So this is our second step, was to take into account to the umbrella sampling, meaning that we bias both the angle and the distance. And we realize that at this stage we have a real peculiar results, because we realize that the scenario is much more complex. There is no possibility to approach, although we can try to go in the groove in the top and upside in the bottom, but at the same time the barrier is too high. On the contrary, when we take into account a nucleotide that's reactive, we observe that different minima observed, different binding modes are possible, and this based on the fact that there is specific angles of approach that at the attack that are important. So this brings us then to look at the reaction. And to do reaction, we look at the economic calculations. At first, we look at the distance profile without taking account of the full chemical reaction to investigate the effect of orientation. And to do so, we consider the reactive region of one nucleoside, and the rest of RNA is taken account in another region to thus take into account the conformational changes. And so I want to also to tell you and remember you that in this kind of simulations, we have different types of coupling that can occur with QMM and memory regions that are quite crucial in our computations. This part, I hope that I show you that different shape reaction and reactivities can take place. Different geometrical parameters can correlate to shape reactivity, so it's a multifactor problem. And we have also the PMF that allows to identify and highlight the reactive formation of dependence on the sides of the problem. I didn't have time to show you that, but also this is quite important. You also observe the importance of specific interaction to form reactive conformations, and also we computed the first QMM calculation of shape probes. What's next? But what next is really important because we want to improve, because I didn't have the time to show you, but we use a very low level of QMM calculation, means that we didn't take into account really precise calculation, but we wanted to do more, and then we also want to take into account the full reaction, chemical reaction at this place. We want to send our work on shape probes to our systems and to other probes, and we wanted to better characterize the reaction and local environments using some geometrical parameters, as well to set up a new computational approach to go beyond the QMM calculations. So now I just want to show you two results that I have on how to take into account conventional changes, since these are quite important in RNA. I hope I convinced you in the beginning, and to do so we can do normal mode analysis. And instead of using Cartesian coordinate normal modes, that is the more standard approach, I developed and used internal normal mode analysis, meaning that instead of taking account the x, y, z coordinate of the atoms, I take into account the angles, the bonds, the distances, and this reduces a lot the number of variables and the computational time. So to do so, I consider mostly the torsions on my work on the backbone of the sugar, and I can do that in an atom on a cross-grain on different representations. And this is quite nice because we have, although the equation we have to solve is a generalized eigenvector problem that is a bit different from the standard approach, it allows us to have immediate information what are the most important variables that undergoes the conformational changes. So we demonstrated that thanks to our approach, we can capture the most flexible region of RNA. And so this is a bit, it's quite nice because the techniques takes like a couple of seconds to be computed. And also we are capable on one side not to deform RNA, the bonds, but at the same time to predict the that are comparable with experiments. And to do so also test and challenge our method and look at a specific virus RS, that is a specific important element that is recruited from, we wanted to understand that to avoid the recruitment from the ribosome. And this with large conformational change can occur when it's recruited with the correct paravirus. And we observed that we were capable to show the most functional movement and also possibility of breaking hydrogen bonds that are not possible in mass simulation. So this for us opened the route to use this approach in integrative approaches like use the SACS, REM, or other data. So to go further, we wanted to also investigate protein-RNA complexes and interfaces, and to do so, why we do so? Because RNA and protein-RNA complexes are also crucial for any cellular function, but until now, when we look at these systems, we look at the document experiments and PDB structures, and we realize that when you look at that, we have a static view. We don't take into account the interface and the dynamics, and on the contrary, recent works, also from us, show that the interfaces are really dynamic, also for the protein, so it should be also for protein-RNA structures. So we did that by looking at different systems, Elisa SARS-CoV-2, in collaborations with the group of Dina Rossa and Sarah Harris in the UK, the pre-5S maturation by M5 enzyme, and finally, what we want shown today is systematic investigation of protein interfaces and dynamics. So to do so, we take in account, we decide a benchmark of different RNA protein complexes with different sites, with different interfaces, with different properties, and different also biological function. And we compute up to 1.5 microsecond atomic simulations. And these systems, what we did was to set up a protocol to investigate protein interfaces. And to do so, we look at the joint property of the interface, conformational changes of binding, RNA characterization, water-mediated contacts, and even more clustering based on the interface. So regarding conformational changes upon binding, we compare now two cases that can occur. We compute a principal component analysis based on the simulation of the complex. And we wanted to, for example, look at what kind of changes occur, if there are more local changes or more rigid body movements. And here it's some pretty nice example. On the left, the ribotoxin restrictor srDNA shows that although in the bound protein RNA there is mostly no changes, there is changes at the level complex, meaning that it should be a more rigid body movement. On the contrary, for nucleolin pre-RNA, we have changes of the bound structure of RNA and the protein. So the changes of the RNA binding site is more due to all of this of local and global changes. And if you look at this last complex, we also analyze the change of RMSF, so root mean square fluctuation, along with simulation for the bound and case. And what you're saying that from one side, the RNA is flexibilized when it's in the protein, and the protein is redefined. So meaning that this should be somehow an effect, or maybe has also biological importance to allow the RNA to make its function. So we also classify our structure, because when we look at the contacts along with simulation, we realize that there are systems where the contacts are really stable along all the simulation time, but in our case, they changes, they decrease, and then they are stable. In our cases, on the contrary, there is a decrease, an increase, and looking at we're like two states. And to do so, we decide to compute, but we compute for each snapshot, each time the contact matrix. I like here an example how we put that, but contact matrix we cannot put in for each time, we cannot put in a plot. So we decide to compute the Jacquard index. The Jacquard index is equal to the ratio between the intersection between these two contact matrix and their sum and their union. Meaning that if Jacquard index is equal to 1, meaning that all the contact matrix equal and 0, they are completely different. And this is an example how to look at it. And now we need to classify this Jacquard matrix. And to do so, it's quite complicated because we have different parameters that we have to take into account, like the cluster variance on one side, and that can give you some information. We want that cluster variance intercluster is stable, and the other side we wanted also to project the different principal components, our results, our points, and we observe that, for example, for this specific complex, the scenario is quite, we have a large variability, but also we have the different clusters are very far away from each other. So to summarize our results, we obtained for based on the different clusters, or different clusters were obtained for different complexes, and there was no dependencies on the size of interface. On the contrary, we found that for several cases we had one substate, about other we are at just up to 5 substates. And these substates have different properties and different biological, in our opinion, relevance. So on this complex, we can also, when we have more than one state, we also compute the variance of the concept along the substate, and we observe, for example, this always for this complex, that there is some region of RNA where the variance is very high between complexes, meaning that different concepts occurs in the different cluster. And we also look at the packaging, since we shown outcome at the beginning of my presentation that is important for RNA to be reactive. And we realized also that when the protein interact with RNA, there is a change of packaging from C3 primes to C2 primes, meaning that maybe we can also use this kind of piece of information to try to have also information at the shape data at this level to have new insights on the structure. And also, based on our cluster analysis, we obtained that this cluster has a different packaging, meaning that we maybe have also different functions. And just one last slide of this result, that we also look at how there is the communication between the different clusters and based on the different interfaces. So we use the generalized correlation-based dynamic network analysis based on the program DNETA. And this allows you, for example, to determine different properties like the communities. The communities are the regions that are together and find the dynamics, so they move all together. At the same time, we can also compute the minimum path, the shortest path, and to do so in this algorithm, we use a Floyd-Warshall algorithm. And this was pretty nice because we decided to look on beside of our works and our groups, we decided to take into account of the different clusters to see what's going on. And we realized at the first stage that different communities are present. And even more, we realized that communication change based on the cluster. In the first cluster, for example, we have a communication that goes through the loop of the RNA between the two domains of the protein. On the contrary, on the cluster 4 here, and it was the similar result of our cluster, there is a communication that also includes the stem, that in this specific RNA is important and was claimed to have a specific biological role. So meaning that by divide the simulations and to find these different substates, we can in principle also try to have insights on the different story based on the different also function and different also properties of the dynamics of this interface, that usually when you think about, again, the structure is just one structure when you take from PDB. So to conclude, I hope I convinced you that our capability to describe our larger RNA conformations change using iNMA, normal homogenized internal coordinates, to have set up a specific to identify alternative interfaces, and we also identify key special next. Now it's use iNMA, so I have discussed that with people and analyze and apply our methods to large protein-RNA assemblies. So this was not only by me. I have people that were involved, team, computational team, my collaborators, the findings, of course, and you for your kind attention.
Thank you for the talk, Dr. Frezza. It was a wonderful one. Now we can continue with our Q&A session. If you would like to ask a question, please write to the chat so I can read out the message. Thank you for the fantastic talk. While you were investigating the protein-RNA interfaces, I think you performed only single approximate 1 nanosecond. Sorry, I don't know.
Yeah, I read the question, yes, so I can answer the question. It's really, so you're true, you are right. We use just 1 microsecond simulations, but so this was a part of my systematic study, but we also did other studies in the past on early cases, and we run several replica, I don't remember how many, with different force fields, and we observed similar results. We also use enhanced sampling techniques for the pre-5S maturation. I didn't have the time to show the results. And so what we think it is important that although the simulations are short, we try, first of all, it was the first time that people look at the dynamics of interface from protein RNAs, because people usually look at just a snapshot. And we also look at the different, for example, for each complex, we look at what was written, for example, for the experimental paper, and also for what was biological, for example, for the system, there was some biological data that showed that the stem has an importance, but we couldn't obtain that from experiments. So we thought that we obtain with our pathway is very short substate to have the full characterization with very long MD simulations. But this was really for us a way to say this approach, it works. Our approach is quite important. And we wanted now to really do a large investigation and to see, also there's another thing that I didn't mention, is that protein-RNA in force field can be also a bit tricky. And so we also, for example, for the LEK study, we use different force field to see also the coherence of results. And for that, for example, the story of a ring that it seems no one thought about before. And it seems that there is a we can find that all our simulations. And I can maybe exaggerate a bit, but I'm not saying so. I have mostly 20 or 25 simulations and even more for complexes. And all the time I see that. So this, I think, is okay. Yeah. So no, it doesn't depend on the clustering side. So we did both of that. We do the clustering. So we did a rough pathway analysis. This is not, I didn't write this code, but I used for one this literature because it's the only one, for my knowledge, that can be applied without any doubt for RNA, without make approximation. And we do the allosteric analysis on all the trajectory. And I divide them into clusters. And also we can use this approach to separate the different windows and different regions. And we saw that was merged together in the full trajectory. But we saw the different pathway, but sometimes were together. And so with the reason why we decided to separate. Also, I want to add that because I didn't do that at the first stage of my paper. It was my reviewer that asked us to look at pathway on the different interfaces. And so, as we know that it's sometimes we complain about reviewers' comments. And at the end, I thought that this was really a nice comment that he made to us. We did a lot of work to try to figure out that. But at the end, it allows us to have a really nice, much more nice paper and a more clear also explanation, because at the end, the fact that with no cluster and sites also dependence, because we also try to stripe our results and we didn't see nothing was changed. Also, what is nice, I didn't show here, you read the paper, but we can also decide with this technique, for example, to look at what's called the bitterness between the different paths, different nodes. And we can see also when several pathways are following the same nodes, meaning that this is a more robust also analysis that we can make. I read the letter. So were any way. Yes, it helped a lot to be a dynamic network. At the beginning, we didn't put it because we wanted it to, it was a massive work. We started this work in 2020 and we published in 2024. So just to give an idea how much time it sometimes takes for science to have some results that are meaningful also. Is there any way to find the specific interaction between transcriptor for DNA? I'm trying to do docking first, and I'm trying to simulate some alarm simulation. So, is there any way to find the specific interaction between transcriptor factors and DNA? I'm trying to do protein DNA docking first, and then I'm trying to simulate full atom simulation. In between, we use the PMFs. Is there any better way to predict this specific interaction? So it's a really tricky question. So I did a bit of work on protein DNA and transcriptor factors in my past. And I think that also, so first of all, the docking can also have issues because you have to be really careful when you do all atom simulations, because you can easily lose your interactions. And PMF also can be tricky because you can do it two ways, either by dissociating or by associating. I think for the prediction of specific interactions, so it was developed in my lab, is a technique we call attract that you can try. I didn't develop, but it was in my postdoc lab, from the library developed. And I think that can be useful. And there is also one paper that was published. But what also in this case people did was to do this type of analysis of, we discussed at that time to do the content analysis at first, because the content analysis allows also to see if you have some weird results also. If what you get is some, also to verify that your contacts also for DNA is even more crucial, because you should be sure about your hydrogen bonds, and you have to characterize that. And sometimes it depends also on principal factors and your system you start with. You can also some hand effect. So you'll be really careful to see if your results are not affected by that.
Thank you for answering the questions, Frezza.
Thank you very much to invite me. I hope that you appreciate it, and thank you again. And I wish you a really nice and excellent evening.
Excellent. So,
bye.
Thank you for joining the session. Goodbye, everyone.